There are multiple ways that you can diagnose malaria. The quickest way is to use a rapid diagnostic test, which has the protein histidine-rich protein 2 as the antigen. It's not the most sensitive way of detecting malaria, so you need quite a high load to pick it up. And also some of the parasites, due to polymorphisms, may not have this histidine-rich proteins 2 that you're looking for, and so you may think it's not there when it actually is. So the gold standard for diagnosing is actually microscopy. For more sensitive detection, polymerase chain reaction is the best, but for routine purposes, labs would use microscopy as the gold standard. So inside the red blood cell, RBC, the parasite progresses through several stages. So there's the early ring stage, which may also be referred to as the early trophozoid. There's the trophozoid proper stage. Then there's the schizont, which has multiple nuclei. And this is the stage that ruptures to release more parasites. And it's in the form, at this point, when it's released, it's a merozoite. And so it's all this rupturing too, keep in mind, that causes anemia of the patient. So here's a visual of the stages that it goes through. So the final stage after the schizont and eruption is, in a small minority of cases, you will get gametocytes forming. These are the sexual forms that will stay in the blood until a mosquito bites the patient and takes it with it. And in the midgut of the mosquito, it will develop through the oocysts and ookinete and form a sporozoite, which can then be brought back to a human when the mosquito bites a human. So this image credit goes to microbeonline.com. So under stress conditions, this is when you get the gametocytes forming. And so as mentioned, when the mosquito bites for a blood meal, and so the blood meal is really the reason why you get bitten. And the blood meal is because the female Anopheles mosquito actually requires the hemoglobin for development of its young. So in the laboratory, in clinical laboratories or research laboratories even, you will prepare smears if you want to visualize it. In the research laboratory, you typically would only use a thin smear so you can really get the details and look at the species that you have in your sample. But in the clinic, 2 types of films are prepared. There's a thick film. The thick film is used for detecting and quantifying the parasites. So the thick film here, the aim is to actually lyse your red blood cells, and they lyse during the washing stage after Giemsa. Because you don't fix this film, you tend to get the lysis happening as soon as water, the red blood cells come in contact with water during the washing stage. The parasites will appear grainy. So your white blood cells are what you use to quantify against. So because you don't have the red blood cells because they've burst, you will use the presence of the white blood cells as your benchmark for how many parasites you have in terms of reporting. And we'll be looking at the calculations or the quantification metrics that are standardized for reporting parasite load in a thick film. So here's an image of a thick film for P. falciparum. As you can see, you get these chromatin dots that are appearing, and you get very thin cytoplasm showing through, but mostly all you will see in the thick smear are these dense chromatin dots. And these images are all going to be from the CDC from now on. And so what you notice is the absence of the red blood cell in your thick smear. If we have a look at a thin smear on the other hand, so the thin smears, what you typically see is that you have red blood cells. They will be intact because what you do is you actually fix them with methanol. And here you're able to see the morphology and the cell deformation. And morphology and deformation are actually important for speciating the Plasmodium parasites, because in some cases the morphology of the red blood cell changes as we have a look, whereas in other cases the morphology doesn't. So it helps, and so having it also provides structural detail of both parasites and infected red blood cells. So here's a look at what would be referred to as a ring stage or early trophozoite. And so what you see are these darkened chromatin dots of P. falciparum as it would appear in a thin smear. And in this, you're really interested in the parasites inside the red blood cell. You don't really care about the ones that may be outside, so that you can speciate as well as take note of the parasitemia, which is the number of parasites in the red blood cell. So we've talked about preparing these smears. How do you actually do it? So you can do it by two ways. You can collect the blood via capillaries, so this is your finger prick, or you can do the venous blood collection where you actually put a needle into the vein and collect the blood. And so if you're preparing a thick film smear, you're going to use 6 microliters of the blood, but if you're doing a thin, then you use 2 microliters. The thick film is designed it's not to be fixed as mentioned, and it's designed to really give you an idea of the quantity, how much parasitemia you've got. The volumes are calibrated so that you actually get the right density. So your 6 microliters of blood for your thick film should give you a semi-transparent smear, while the thin smear allows for more accurate speciation because it's much thinner. So just 2 microliters of blood is used. And so how you fix your thin films, as we mentioned, you don't fix the thick one, is that you dip it or you spray with methanol for just a second. And this essentially glues the cells to the slide. So you don't want to go more than a second, because if you do, then the fumes or the volatility of the methanol can actually fix the thick smear. So what happens is you actually use the same slide for the thick and the thin. So you do the thick first, and the thin you're going to dip or spray the thin. And so if you don't do it quickly, then you can actually get the methanol traveling up and fixing the thick smear, which you don't want, because you want the red blood cells to burst, releasing the parasites. So what do we use for staining? You're going to be using, after you fixed it for just a second, you let it dry flat so that everything will be fixed. So your thick films and your thin films are going to be, what you use is a stain called Giemsa or Giemsa, I'm not sure how it's pronounced, and you use this stain is a mixture of methylene blue, which is a basic dye, and eosin. Eosin is an acidic dye. So for hospitals, they typically, because of need for speed, they will do a 10% solution for rapid diagnostic work, and here you stain it for 10 minutes. But for research, or if you really want detailed speciation information, you will do a 3% Giemsa stain. So this gives you higher quality staining for your slides. And here you will leave it for about 30 to 45 minutes for the 3% stain to really infuse into the red blood cells, into the parasite, and stain the right organelles for visualization. The slides, once you've made them, don't have to be read straight away. Okay, so once you make your slides, you don't have to stain them straight away. You can just let within 72 hours. As long as you do it within 72 hours, you will get appropriate details to speciate your parasite or count your parasite if it's a thick smear. It's important to mention that the Giemsa stain is made with buffered saline, buffered water. So you use buffered reagent, phosphate buffered saline at a pH of 7.2 for preparation of the Giemsa. You really need this because it helps the dyes to bind to the right organelles of the parasite. So for instance, the eosin, being acidic, will actually go and bind the chromatin. I know DNA is actually negative, but because of the histones, specifically the lysine and arginine amino acids will make the structure, the overall chromatin, a bit positively charged, so the eosin will go and bind it, whereas the methylene blue, being basic, will bind in the cytoplasm, which is relatively acidic. So let's have a look at how you would tell the different species apart. So for P. falciparum, it's important to know that it infects mature red blood cells. Now, the thing with the fact that they infect mature red blood cells is that because they're already grown, then they don't have capacity to enlarge. So you wouldn't see red blood cells enlarging when P. falciparum infects it. So what you'll see multiple chromatin dots, you see what is referred to as like a nipple-like protrusion, and this is when the chromatin dot is just outside of the cell, right, while the body of the parasite is inside the red blood cell. So the cytoplasm is inside, but then the chromatin may be just outside, giving this nipple appearance. You may see, well, you'll definitely see ring, but you may also see comma shapes. The gametocytes in P. falciparum are banana shaped, and you may also see what's referred to as Murr's dot. So here you see the trophozoite in a thin blood smear. So here's what a schizont would look for P. falciparum. You can see the multinuclei, which are merozoites, lots and lots of merozoites that have developed, that are about to burst and infect other red blood cells. So P. falciparum, you can get up to 32 of these schizons, and so you can see there are lots in that image. Let's have a look at Malaria, P. malariae. So these ones have a characteristic band form or basket form, or what can be referred to as a bird's eye form that they make. They're often seen in the schizon stage in thick film. You will see about 6 to 12 merozoites, usually on average 8 to 10. So they have less than the P. falciparum. And they're easier to identify via the ring stage in the thin film, just because of this banding structure that they form. So here's what a bird's eye form would look like. And this one is a ring form trophozoite in P. malariae, and this one is in the thin film. And you will also see the trophozoite looking like this when it's like a full-blown trophozoite, but at the bottom here you can actually see a ring form trophozoite, which is, you definitely have the ring shape more than the one up on the top left of the screen. And here's another trophozoite, and this time this one is in the thick smear. So you see there's some white blood cells floating around there, the darker, larger structures, and that's just the thin cytoplasm and the chromatin in the center for the trophozoite. And this is the banding form that's characteristic of the P. malariae. So you can just have it going across, or you can have it looking a little lighter, as you see on the screen, like taking up a lot more of the cells of the red blood cell. And finally, here's one that's like a little bit irregular looking as well for the P. malariae trophozoite. And this structure here is again a trophozoite from P. malariae, but this one will be referred to as a basket form. I guess you can imagine the handle and the basket right there. So here's a gametocyte of P. malariae. You can see it's not banana shaped, it's quite round. And this one is in a thick smear. But if you have a look at the thin smear, you can really see that it takes up a large part of the red blood cell. And here's another one with a bit more of a lighter background. So it may appear like that. And here it's in a thin smear once again. So here's the schizon stage for the P. malariae in a thick smear. So you can see that it has less merozoites or nuclei that you would see in a thick smear compared to the P. falciparum. So you're expecting about usually 8 to 10, but you can get up to 12 of these merozoites. And so that's what it looks like on a thin smear, where you can get a bit more detail. You can see that the nuclei here, or the dots, are a lot larger than what we'll be looking at in a little while when we look at like an ovale schizont. Okay, so let's have a look at the P. ovale, what it will look like. So I've got a little chick coming out of the egg, because so P. ovale likes to infect young, immature red blood cells called reticulocytes. And another thing that differentiates it from what we've discussed, the oval, is that, the cells become oval and display these spiky edges called fimbriation. They sometimes also display a red zone in thick films. So this is what it's looking like in a thick film for P. ovale. Got the chromatin dot and I guess some red bits around. I need to look at a lot more of these before I can feel comfortable at identifying them. But so here's your ovale ring form trophozoite, as seen on a thick smear, You can see the thinner cytoplasm and then the chromatin in the center. So a bit more of a close-up. And here's a cell that has multiple ring-formed trophozoites. So you can see with the ring form, they do form that shape. One has a lot of rings, whereas the other has just the one, the other red blood cell on the screen. And this one is a thin smear. And here is more of the ovale ring form trophozoites here. Can you tell any differences between the ovale ring form and the P. falciparum? To me, yes, after experience, maybe you'll be able to tell at a glance, but it's not too obvious to me. I can see that we tend to have a lot more rings in the infected red blood cell, and actually that's apparently a feature, so you can have multiple infected red blood cells. I think you can have it with Plasmodium falciparum as well, though. But the fimbriation should give it away. So you've got the cell with kind of like this edgy, zigzaggy structure. So you'll get the fimbriation happening. And here's another fimbriation feature. So it just kind of looks like the edges have been ripped off a little bit, splattered look. So the schizons for P. ovale are quite interesting. They're quite large. So in a thick smear, to me, they look a bit similar to what I thought was in the P. falciparum stain, for instance, but this is actually the ovale cell. And here's another thick smear of an ovale infection in a cell. So here's a schizont in a thin smear, so you can get a bit more details. You can see the multiple nuclei, which would be the merozoites. But what you can see here with this image is that it is smaller because this is a thin smear. And if you remember the P. falciparum schizonts, they were larger, and these ones are slightly smaller. So ovale, I think the feature is that they're smaller. So here's a gametocyte for P. ovale. You can see it's definitely not banana shaped. It's quite large, and I think this one here is referred to as a macro gametocyte because it's quite large. And I'll show you the micro gametocyte. You can see it's slightly smaller than the macro. But in the thick smears, I found them a bit confusing on the thick smear. Like, to me, that looks like a schizont rather than a gametocyte. But it is a gametocyte. And here's another gametocyte in a thick smear. There's no way I would have picked that up as a gametocyte, but I'm new to this, so people that are experienced will be able to pick it. Be strong. So let's look at vivax. So vivax tends to have these enlarged amoeboid shapes. The schizons are large and visible in the thin film. Well, that's relatively larger. So the gametocytes are round, unlike the banana shape of the falciparum. So these ones also infect reticulocytes, and that's why I've got a baby bottle next to it for association. So you've got your vivax ring-formed trophozoites in this thick smear. I really don't think I would have picked this at all in a thick smear. So here we have more ring-formed trophozoites in a thick smear for vivax. I may have thought it was cell debris. And here we're having the trophozoid ring form in a thin. So in a thin smear, you start to get way more detail. And I can see that while it's a ring, it's still not, I could differentiate that potentially from a falciparum or malaria ring, because it seems to be not like as perfect a ring. And here are more of the vivax ring form trophozoites in a thin smear. But these ones now we're going to look at, I think this will be the characteristic thing for vivax, is the amoeboid appearance in the trophozoite stage. So you can see it's quite round at the trophozoite stage, and it's quite large. So this one here is the thick smear, while this is the thin smear. So let's look at the count for these smears. So quantifying parasitemia helps determine disease severity and treatment efficacy. For your thick film counting, you want to count parasites relative to 200 white blood cells. And again, the white blood cells, because you would have burst the red blood cells. So you use two tally counters, one for the parasite and one for your white blood cells. If you count less than 100 parasites after counting 200 white blood cells, then you need to extend your count to 500 white blood cells, because you're really trying to have at least 100 or more parasites to make the count valid. You use your estimated white blood cell count as 8,000 white blood cells per microliter. But if you have a machine that can actually assess the full blood count for you so that you get the actual white blood cell content, then do that. But if you don't, then you can just estimate it to be 8,000 white blood cells per microliter. So the formula that we use in a thick film then is, if you counted more than 100 parasites, then you have the number of parasites that you've counted, you divide it by 200 white blood cells, and then you're multiplying it by the 800 expected white blood cells per 8,000, I should say, 8,000 expected white blood cells per microliter. In the instance that you counted less than, and then you're forced to count up to 500 white blood cells, then the formula is the number of cells, parasite cells that you've counted, divided this time by 500 white blood cells, and then multiply it by 8,000 white blood cells. For the thin films, you're going to want to count at least 2,000 red blood cells, because this time we want to see the parasite inside the red blood cell. Then the formula you're using is the parasites that have infected red blood cells. You divide it by the 2,000 total red blood cells that you've counted, and you're multiplying it by 5 million red blood cells, because you're expecting about 5 million red blood cells per microliter of blood. You will have to use your thin films to actually count your parasites when your thick films show greater than 100 parasites per field, because then you can't really count accurately. Importantly, before you can declare a slide as negative, you have to have counted at least 100 fields. For those of us that work in parasite culture rather than in diagnosis, as in a hospital, we have to count at least 250 red blood cells per field, and we need a minimum of 1,000 red blood cells to make the count valid. And so then we use our results to determine percent parasitemia. So as you can see, microscopy offers a robust, low-cost, and effective way to diagnose malaria and monitor treatment success. So mastery of smear preparation, staining, and species identification is essential for accurate diagnosis and public health monitoring. Thank you for joining me on this video. If you enjoyed it, you may also want to check out the parasite life cycle, which has already been covered, or you may also want to check out a run through of Plasmodium as a group in general. They're very unique creatures. So thank you once again. Please subscribe, give it a thumbs up if it was helpful, and I will catch you in the next one. God bless you.